All right, so these initiation steps have brought us to this stage where we now have a complete ADS ribosome at the initiation codon. We now have a met tRNA positioned in the P site. And now the next step, of course, would be to put in the next amino acid in the A site to start protein synthesis. Viruses, again, do this differently. Some viruses, they can initiate translation without methionine. Of course, that's called methionine-independent initiation. And here in panel A is one example of this. This is the iris of the insect picornavirus, cricket paralysis virus. As the name implies, this is a virus that paralyzes crickets. And it's very related to polio, just like polio paralyzes humans. This virus paralyzes crickets. And it initiates translation without a methionine. And how it does so is shown here. The iris of the viral RNA, so the viral RNA is shown in green here, it folds to mimic an initiator methionine tRNA. You can see this RNA is folding back on itself. It's base pairing with itself as if it were a tRNA, and it's sitting in the P site of the ribosome. And the ribosome thinks it's a initiator methionine tRNA. So it says, we're ready to go. We're ready to make proteins. And the next thing that happens is the insertion of the right tRNA in the A site. And so the protein actually will begin with whatever that next amino acid is, and not a methionine. It's really quite remarkable. Here's another example of that. This is a plant viral RNA. So this is a positive strand RNA virus of plants. Here's the 5' end. It's got a cap. 3' end forms a tRNA-like structure that sits into the P site and mimics the initiator tRNA. And this tRNA-like structure happens to have a valine attached to it. And so that the protein encoded by this viral RNA initiates with a valine and not a methionine. This is interesting because the way we figure out how many genes are in any organism once we get its sequence is by looking for open reading frames that begin with a methionine. So we may be underestimating the number of proteins encoded in any genome if we just do that because we're missing these methionine-independent initiation events. Let's look a little bit more at the genome of this unusual cricket paralysis virus. It's shown here. This is a plus-strand RNA virus, just like poliovirus. It has VPG at the 5' end. It's polyadenylated. One of the main differences is that the open reading frame is split into 2 open reading frames in cricket paralysis virus. As you recall, poliovirus and other picornas have a single open reading frame. This one for this insect virus is split. There are 2 irises in this genome. There's one at the 5' end, shown down here, to allow internal initiation in the absence of a cap. Remember, these don't have caps, these genomes. And then there's an iris in this intergenic region between the 2 open reading frames. And that iris is the one in which the RNA folds to mimic an initiator tRNA. Now, you need an iris here between the 2 open reading frames because, well, actually, if you made a single polyprotein, you could process it to form all the proteins you needed. But apparently the virus has chosen to make 2 polyproteins. And to get this second one translated, you have to have an iris here because ribosomes do not efficiently reinitiate after termination of the first open reading frame. So this brings us to a thorny issue in translation of eukaryotic viruses. So this is illustrated on this slide. On the top is a typical eukaryotic mRNA that we've been talking about with a cap and 5' and 3' untranslated regions, a poly A tail, and a single open reading frame. And this is the way most eukaryotic messages are constructed. They are monocistronic. They can only encode one protein. And that's in contrast to bacterial and archaeal mRNAs, which are polycistronic. You can see here these mRNAs have multiple open reading frames in them, so that ribosomes can initiate translation at the first AUG, then they terminate, and then ribosomes can initiate at the second and terminate, the third and terminate, and so forth. You can make multiple proteins. And the ability to do this is because there are Schindel-Garno sequences around the AUG, which are complementary to ribosomal RNA sequences, and they allow the ribosomes to bind at each AUG. It's sort of like an iris, but very different mechanistically. Eukaryotic cells don't have that. Now, you may ask, why not? You just told us that there are iris sequences that allow internal initiation. So in theory, you should be able to construct this kind of an mRNA in a eukaryotic cell, and by putting an iris in between each open reading frame. And we know these work because we construct them in the lab. People make dicistronic mRNAs all the time for production of two proteins in eukaryotic cells. But there are no naturally occurring ones as far as we can tell. There are no naturally occurring eukaryotic polycistronic mRNAs where the iris is separating the open reading frames. I don't know why. We don't know. Now, this monocistronic restriction is a problem for viruses because, as you remember, many viruses, their genome is a single mRNA. So how can they make multiple proteins? So that is the conundrum we're going to address next.